programang ito ay rated SPG. Striktong patnubay at gabay ng magulang ang kailangan. Maaaring may masiselang tema, lengguwahe, karahasan, sexual, horror o droga na hindi angkop sa mga bata.
Now we are proceeding to the confirmatory testing or microscopic examination. Here are the things that we will be using to conduct a confirmatory testing or microscopic examination. The evidence bag, sample, forceps, surgical scissors, dropper, and our saline solution. Here are the process of conducting confirmatory testing or microscopic examination. Using a surgical scissor, we will cut some part of cloth that contains the suspected seminal stain. After cutting, we will put it to the beaker with our saline solution. The purpose of this is to separate the suspected seminal stain from the cloth. And using a dropper, we will take some samples and put it onto the glass slide. This is the appearance of a sample under a microscope. As we can see, we have the head and the tail of a sperm separate from each other. Now we proceed to the part 4, the analysis. The tailless problem. Yes, in other stains, it is very common to observe only the heads of sperm cells without the tails. The reason is the tail is a delicate structure that is easily detached from the head due to the process of decomposition, drying, or physical handling of the stain over time. To ensure the negative control stayed negative, these are the steps that we follow. The use of sterile tools. All instruments, slides, and containers were clean and unused to ensure no foreign materials was introduced. The proper handling. We avoided touching the area where the sample was placed with bare hands. The separation. The negative control was handled separately and placed far from the actual samples to prevent accidental transfer of substances. Now, the conclusion. Based on the result of the acid phosphate test and microscopic examination, we concluded that the sample collected is positive for seminal fluid. The AP test served as the presumptive screening method indicated by the appearance of color, which is purple. However, it is important to note that the AP test alone is not conclusive, as other substances or contaminants may be produced similar reaction leading to a false positive. Therefore, microscopic examination remains the definitive method to confirm the presence of the semen. The family of the suspect is not quite satisfied with the result of the microscopic examination confirming the presence of seminal fluid, and then requested the DNA analysis to confirm if the semen is really came from Mr. Boncha.